Good morning, students. Myself, I'm Dr. Indira, dealing the course Probiotics and Functional Foods, Module 1, Unit 1. Today, we will see the topic that is the prebiotics and gut microflora. So, in the previous topics, we have seen the probiotics and their health benefits and how to characterize the probiotic strain and how to manufacture and how to formulate the probiotic products and what is the minimum effective dose of a particular product and how to store or maintenance the probiotic microorganisms. All these topics we have discussed earlier. Now we will see the prebiotics, gut microflora, and its health benefits. So probiotics are nothing but live microorganisms which when administered into the host, they confer health benefits. Come to prebiotics. Prebiotics are different. Probiotics are the live microorganisms, whereas the prebiotics are the non-living substances. So here the concept of prebiotic was first introduced by Glenn Gibson and Marcel Roberford. These scientists, they explained that prebiotics are the non-digestible food ingredients that selectively stimulate the growth or activity of one or a limited number of bacteria in the colon. So prebiotics are the non-living substances, nothing but the non-digestible food ingredients. These ingredients stimulates the growth of the bacteria, nothing but the probiotics in the intestine. So later in 2004, Glenn R. Gibson and Holly M. Probert proposed three criteria. One is the resisting hydrolysis by mammalian enzymes and gastrointestinal absorption and gastric acidity. And then second one is fermented by gut microorganisms. And third one is selectively stimulating the activity and growth of gut bacteria involved in health and well-being. So these are the different features of the prebiotics. One is resisting the hydrolysis of mammalian enzymes and gastrointestinal absorption and acidity. So these prebiotics, they resist these enzymes as well as the acidity and fermented by gut microorganisms and stimulate the activity of the other beneficial bacteria, nothing but probiotics. So these are the features of the prebiotics. So in 2016, the International Scientific Association for Probiotics and Prebiotics, they redefined that prebiotics as a selectively utilized substrate by host microorganisms conferring the health benefits. So these are called as the prebiotics. We'll see what are the different categories of prebiotics. First one is the fructans. Fructans are the natural fructose polymers used for their prebiotic and health enhancing properties, particularly in the functional foods. Unlike starch, these fructans are derived from plants, which are water-soluble compounds derived directly from the sucrose. And if you see the structure of these fructans, they carry the inulin beta 2 1 linkage and 11 fructan has beta 2 6 linkage and graminin has beta 2 1 linkage and beta 2 to 6 linkages. So these are the examples of the fructans. Differences in the structure arise based on the position of fructose addition, leading to the creation of 1 kestrose, 6 kestrose, or 6G kestrose. So if you see inulin, it is a type of fructan, it's a polymer. So linked alpha linked glucose and beta 2 1 linked fructose, and this is the composition of the inulin and with longer chains. So with longer chains and oligofructose and fructooligosaccharides with shorter chains. This is the structure of the inulin. In addition, neoinulin and neolevin types of fructans with internal glucose residues can also be found in the prebiotic category. So if you see the benefits of these fructans include, they relieve the gastrointestinal disorder symptoms and increased calcium bioavailability is there, induction of apoptotic effects on the colon cancer tumor cells, reduction of cholesterol and triglycerides, and modulation of the immune system. So these are the examples of inulin and the living structures. And next, fructans. This is the overall other prebiotics and their health benefits on the host system, gastrointestinal system. If you see the inulin, it relieves the constipation reduces the risk of gastrointestinal disease, subjective mood and cognitive performance will be improved, absorption of calcium, magnesium, and iron, and then blood lipogenesis, plasma triglycerol concentrations will be reduced. So these are the properties of the inulin. The other prebiotics we'll see. And if you see the other features of this inulin, it is a water-soluble storage polysaccharide, belongs to group of non-digestible carbohydrates called fructans. And inulin is GRAS status that is generally recognized as safe according to the United States, and it is abundant in 36,000 plant species, particularly with chicory root being the most concentrated source of the inulin. In addition to this chicory root, Jerusalem artichokes, dahlia tubers, vacon, asparagus, and leeks are the natural sources of inulin. So it contains 2 to 1 beta D fructopyranose units, typically with 1 to 2 alpha D glucopyranose group, a hydrolyzed form of inulin known as orgofructose. Inulin is a plant carbohydrate stored with a fructose motif connected by a beta 2,1-D fructosyl linkage. So it is not digestible in the human small intestine because of its beta configuration of anomeric C2 carbon 2, but it can be fermented in the large intestine. Almost 90% of inulin travels to the colon where the bacteria digest it there. So if you see the health benefits of this inulin, it reduces the blood lipogenesis and plasma triglycerol concentrations, relieving constipation, reducing the risk of gastrointestinal disease, and enhancing the absorption of calcium, magnesium, and iron. And next, fructooligosaccharides. It's a common name for fructose oligomers, generally understood as inulin type oligosaccharides. They are a series of homologous oligosaccharides derived from sucrose, usually denoted by the chemical formula GFN, consisting of 1 ketose, nistose, and 1 fructobeta, fructopyranose nistose with 2, 3, and 4 fructosyl units bonded to the beta 2,1 position of glucose, respectively. In general, these fructooligosaccharides occur naturally in some vegetables such as onions, beet, rice, shallots, tomatoes, and bananas. Or it can be produced from sucrose or inulin by microbial enzymes, namely beta-D fructopyranose or fructonyl transferase, such as bacterial and fungal sources. Fructooligosaccharides consumption shows to prevent colon cancer, immunomodulatory effects, control and manage obesity and diabetes, improve mineral absorption, and regulate the serum lipid and cholesterol concentrations. Come to the galacto-oligosaccharides. These are also non-digestible carbohydrates composed of 3 to 10 or more galactose molecules and a terminal glucose molecule. They are produced through the catalysis of glycoside hydrolases, typically with lactose as a substrate, leading to a blend of galacto-oligosaccharides with varying polymerization levels. Several microbial glycoside hydrolases have been utilized for their manufacturing, whereas biotechnological technologies such as immobilizing or recombinant enzymes have enhanced the production. And these GOS are used in meat-based products and infant formulas to mimic the effect of breast milk oligosaccharides because it is physiologically similar to breast milk. So if you see the benefits of this galacto-oligosaccharides, they are selective stimulation of beneficial microorganisms, reduced production of toxic substances, improved immune response, increased mineral absorption, and reduced severity of obesity and diabetes. Come to the xylogo-oligosaccharides. This is a linear oligosaccharides with D-xylose units linked by beta-1,4 glycosidic bonds and ranges from 2 to 12 and is composed of xylobiose, xylotriose, xylotetrose, xylopentose, xylohexose, and xylohectose. And these are the type of sugars present in this xylo-oligosaccharides. They produce from xylene-containing lignocellulosic materials by chemical methods, a combination of chemical and enzymatic treatments, or direct enzymatic hydrolysis of sensitive substrates, we can produce these xylo-oligosaccharides. To see the health benefits of the xylo-oligosaccharides, they have biflozinic activity, reduce blood cholesterol, maintain gastrointestinal health, increases the mineral absorption, stimulates the immunity, and have antioxidant and anti-cancer activity, and ability to reduce the glucose. Come to the chito-oligosaccharides. These are chitogen oligomers formed by the hydrolysis of chitosan, which contains glycosidic bonds with a degree of polymerization of less than 20 and an average molecular weight of less than 3.9 kDa. So They have more advantages because of their solubility and lower molecular weight, enables the greater biological applications various fields. So they modulate the composition of the gut microbiome uh, by increasing the bacteroids, decreasing the proteobacteria and actinobacteria, and decreasing the firmicutes and bacteroides ratio. So these are the activities of the phyto-oligosaccharides. Come to the lactulose. This is another prebiotic mentioned above. All are plant-derived probiotics, but this is the artificially produced prebiotic. So lactulose is an artificial polysaccharide composed of galactose and fructose generated via the isomerization of lactose. So the methods used for producing this lactulose is the chemical, enzymatic, and electroactivation techniques. So this is resistant to hydrolysis by small intestine disaccharidases enzymes and reaches the colon intact, where bifidobacteria and lactobacillus selectively metabolize it to form the lactic acid, carbon dioxide, and hydrogen gas, and short chain fatty acids, which increases the fecal biomass and decrease the pH. That is the future of the lactulose. These acids, whatever the short chain fatty acids, they are biochemically draw water into the intestine and soften the stools. So lactulose can be used as a laxative. This acidification also promotes the conversion of ammonia to ammonium ion, which is not absorbed and excreted. Come to the another prebiotic, that is the resistant starch. It refers to all starch and starch breakdown products that are not absorbed in the small intestine. Those are considered as a resistant starch. After being digested in the small intestine, starch travels to the colon, where it is fermented by the gut microbiota. While many gut microbiota express alpha amylases that break down the soluble starch, only a small number of gut microbiota can break down resistant starch, which is insoluble and highly resistant to digestion in the small intestine. So the easily digestible starch fermented readily, producing acetate and lactate, whereas the resistant starch fermented much more slowly produces the acetate and butyrate. Resistant starch is categorized into 5 types. That is one, resistant starch 1. This is the starch that is physically enclosed within the cell structure, hindering the enzymatic breakdown. Whereas the resistant starch 2 is the starch is having crystalline structure that is resistant to enzymatic action and loses this resistance when it undergoes gelatinization. In the resistant starch 3, this is the retrograde starch. So it occurs when amylose crystallizes and becomes resistant to enzymes, such as during the cooling of thermally gelatinized starch. Resistant starch 4 is a chemically modified starch that cannot be digested, while resistant starch 5 is a combination of amylose and lipids formed during starch processing. So these are the 5 categories of resistant starch. Resistant starch consumption has been shown to regulate gastrointestinal bacteria, prevent colorectal cancer, and reduce the diabetes and reduce the obesity. Come to the another category of prebiotics. Those are the polyphenols. These are the secondary metabolites of the plants. They range from simple phenolic molecules to complex polymers characterized by an aromatic ring containing one or more hydroxyl groups in their chemical structure. They are categorized into 2 main groups, flavonoids and non-flavonoids. Around 8,000 polyphenols known to exist in the plants, vegetables, and fruits. And if you see these health benefits of polyphenols, the promotion of probiotics, reduction of pathogenic bacteria, which reduces the inflammatory immune responses and decreases the risk of gastroenteritis, colorectal cancer, metabolic syndrome, and inflammatory bowel disease. So these prebiotics, they modulate the gut microflora. Whatever we have discussed, all these prebiotics, they are modulating the gut microflora. Several studies have highlighted the importance of the relation between the prebiotics and gut microbiota. If you see one example, that is increase in the quantity of bifidobacterium and lactobacillus species, which ameliorate the inflammatory bowel illness. It aids in the digestion, resists the constipation, resists the infection, and avoid traveler's diarrhea. It's one of the advantage of the prebiotics. Other studies, if you see, influence the short chain fatty acid production, modulate the immune system, improve the gut barrier function, reduce the pathogenic bacterial populations, improves brain function and mineral bioavailability, and reduces the blood lipid levels and affects the insulin resistance, which is related to the cardiovascular system and more. The fermentation of prebiotics by intestinal microbes produces several metabolites. These are short chain fatty acids. These are essential for balancing the redox equilibrium production in the anaerobic environment of the gut. So these are composed of 1 to 6 carbons, and examples are the acetate, propionate, and butyrate, and they act as a source of energy absorbed through the colon mucosa. The short chain fatty acids have been shown to have many essential physiological functions, includes the maintaining luminal pH, inhibiting the pathogen growth, influencing intestinal motility, and stimulating the cancer cell apoptosis, thereby reducing the colorectal cancer. So this is the way the short chain fatty acids produced by probiotics and produced because of the prebiotic activation and stimulating the growth of the probiotics that produce the short chain fatty acids. So if you see the combination of both prebiotics and probiotics, they regulate the gut microbiome. If you see probiotics, they compete for the colonization sites and nutrients, inhibit the growth of pathogenic microorganisms, and then selectively stimulating the growth and activity of these probiotics by prebiotics. And then these prebiotics triggering the probiotics and which triggers the production of short chain fatty acids. So these improve the gut barrier integrity and then providing the energy. Because of all these, they modulating the immune response. Thank you.